Welcome to the deep dive. Today we're really getting into something that's been buzzing in research circles, a compound called SLU-PP-332. We're drawing from a paper SLU-PP-332, Preclinical Insights and Cautionary Notes. And our plan really is to unpack what makes it exciting, but also, just as importantly, hit those significant warnings that come with anything this early. I mean, this isn't just chemistry, right? It's about potentially tweaking our core energy systems, fat, glucose, even endurance. Sounds almost sci-fi. Let's explore why. That definitely sets the stage. So, okay, let's dive in. What is SLU-PP-332? The source calls it a pan-ERR agonist.
Yeah, that's the term. So ERR stands for estrogen-related receptors. And pan-agonist just means SLU-PP-332 activates all three kinds, alpha, beta, and gamma. Now, what's key here is that these ARRs, they're called orphan nuclear receptors.
Orphan receptors.
Right. They're transcription factors. They control genes involved in energy metabolism. But the orphan part, it means they don't have a known natural key, basically. No known natural ligand. And that's what makes them super interesting drug targets. They're like these untouched biological switches.
Okay, so agonist just means it flips those switches on, it activates them.
Exactly that. It stimulates them, turns them on.
Okay, got it. So we know what it is now. What did it actually do in these early tests? Where did they try it out?
Well, the main studies mentioned were in diet-induced obese mice and also in human muscle cells, but in vitro, so in a dish. And the results, they were pretty compelling, actually. They saw reduced fat.
Reduced fat, okay.
Improved glucose handling, so better blood sugar control. And, get this, enhanced endurance.
Wow, okay. That is quite a combo. Can you maybe elaborate a bit on like the how behind some of that, the mechanism?
Sure. So one big piece involves ERR alpha. It seems to work together with another factor called PGC1 alpha. PGC1 alpha is kind of a master switch for building new mitochondria and making them work better.
Mitochondria, the cell's power plants.
Exactly, the powerhouses. So ERR alpha plus PGC1 alpha essentially boosts mitochondrial function, ramps up energy production. And why should you care about mitochondria? Well, they generate almost all the energy your body uses. Better mitochondria mean better metabolic health, more energy, the whole deal.
That definitely makes sense. You can see the potential implications there. I mean, if that could translate safely to people, oh, wow, but...
There's always a but in early research, isn't there? We absolutely have to talk about the caveats, the risks. Yeah. What does the source warn us about?
Yeah, this is critical. First off, let's talk dosage. It's so important to understand that taking mouse results and applying them to humans, it's really unreliable.
Why is that? Just different biology.
Totally different. Metabolism, receptor sensitivity, how quickly the drug gets cleared, it varies massively between species. You just can't make a direct leap.
Okay, but the source does give the mouse dose. Can you put that number in context? Like, what might that look like for a human? With the giant asterisk that this is not a recommendation. Right, giant asterisk necessary here. So the mice got 50 milligrams per kilogram. If you do a very rough back-of-the-envelope conversion using body surface area, you're looking at maybe around 300 milligrams a day for a 75-kilogram human. But please, nobody should take that as a human dose. It's purely illustrative of the animal study scale. Absolutely not advice.
Understood. Not advice. So beyond just the dosage translation issues, what actual risks did they flag?
Well, a major one is that overactivating ERR gamma specifically might cause cardiac hypertrophy.
Heart enlargement. That sounds serious.
It is. ERR gamma is involved in heart development, and pushing it too hard could lead to unhealthy growth. That's a big concern. Also, at high doses in the animals, they saw elevated liver enzymes. That can be a sign the liver is stressed or even damaged. And the thing is, we have no idea what the toxic levels might be in humans. Zero clue at this point.
Right, and knowing those potential downsides is just as crucial as hearing about the benefits. You need the whole picture. Are there other uncertainties?
Definitely. There are estimates of the half-life, how long it stays active in the body, based on unpublished animal data. Looks pretty short, maybe 1 to 4 hours.
1 to 4 hours. What does a short half-life usually mean for a potential drug? More pills per day?
Yeah, typically it means you'd need more frequent dosing to keep stable levels. That can be a hassle, makes it less practical sometimes, adds complexity. And then there's drug interactions, completely uncertain. So stacking it, taking it with other compounds, extremely risky. We just don't know what could happen.
Stacking sounds like a bad idea then, given the unknowns. Any specific types of interactions to worry about, or is it just anything?
It's pretty much a black box right now. Without proper studies, combining it with anything else is just guessing, and potentially dangerous guessing. Could mess with effectiveness, cause unexpected side effects. Just not worth the risk. Plus, there's another wrinkle. The potency, how strongly it activates each receptor, might vary between alpha, beta, and gamma.
Oh, interesting. So it's not an even effect across the board. Why does that matter?
Well, imagine you have three volume knobs, alpha, beta, gamma. SLU-PP-332 turns them all up. But maybe it cranks alpha to 10, beta to 7, and gamma to 9. That variability makes it harder to predict the precise outcome. Different effects, different potential side effects depending on which receptor is more strongly activated. It adds, another layer of complexity.
So putting all this together, where does SLU-PP-332 actually stand right now in the whole drug development process?
Okay, let's be crystal clear. It's preclinical only. Tested in animals, tested in cells in the lab. That's it. There have been 0 formal clinical trials in humans. None.
And what's the usual journey from this stage?
It's a long one. Typically, you're looking at 10 to 15 years to get from preclinical to an approved drug, maybe longer. And frankly, the failure rate is incredibly high. Most compounds never make it.
So 10, 15 years, high failure rate. For someone hearing the exciting potential, what does that really mean? We talking decades maybe? Or likely never?
It really puts it in perspective, doesn't it? The journey from a lab discovery to something you can actually use is immense. Huge hurdles. So yeah, the vast majority of things that look promising in mice or cells, they fall down later. Maybe they're toxic in humans. Maybe they just don't work. It's a tough process. Exciting early results are just step one.
Which really hammers home the bottom line from the source material, doesn't it? SLU-PP-332. Promising in the lab, yes. But human safety, human efficacy, totally unknown. It's that crucial reminder. Exciting potential is not the same as a ready-to-go therapy. Big difference. So that's our deep dive on SLU-PP-332. You've got this really interesting compound, lots of potential for metabolic health, sure, but it's wrapped in these really significant warnings because it's just so early. And, thinking about things like this, compounds that fiddle with our fundamental energy controls, it does make you wonder, beyond just fixing health problems, what happens when we get really good at this?
Yeah.
At precisely controlling our metabolism? What other questions, maybe ethical ones, pop up when we learn how to truly hack our own biology like that? Definitely something to chew on.